gauge invariant equation of motion for free fields. This is a fundamental equation which we will review later on. But if we want to look at interactions, one can see that it's very hard to write down interactions of mass with higher spin in flat space. And in fact, it appears to be impossible to have a non-trivial matrix for spin greater than 2. And this is essentially because for this higher spin field, due to this gauge invariance, there are associated conserved charges of spin at -1, which should enucleate the S matrix. So there are conserved charges of spin at -1. And these conservation laws for spin greater than 2 are too strong and only allow for a trivial S matrix in flat space. For spin n1 and 2, of course, we know that we can have non-trivial interactions. We have Young-Mills theory and we have Einstein gravity. But for spin greater than 2 in flat space, the existence of these conserved charges only apparently allows for a trivial S matrix. This is essentially a consequence of the Coleman-Mandula theorem. Okay. Can I also use the size of black holes? Okay, so this is in flat space. On the other hand, so people, due to the results, believe that we don't have to worry about higher spin particles because they cannot have non-trivial interactions, so we can only discuss trivial theories with it. But on the other hand, there were some explicit constructions if we go away from flat space and we allow for a non-zero cosmological constant. So if we work in anti-de Sitter space or de Sitter space, there are, if you want, yet no explicit results due to Frapkin and Vasiliev.
Everyone, this is Mr. Sinti, and today I'd like to discuss one of the cool aspects of cell communication, which is signal transduction pathways. That's right. When a cell receives a message, usually in the form of a chemical, it's usually receiving it by some sort of transmembrane protein. Like, for example, in this illustration here, you can see it's a G protein coupled receptor. And so this particular receptor protein will pick up some sort of inducer, some sort of chemical message from the outside. And what will happen is when it receives that message, it'll change shape. And the reason that it's called a G protein coupled receptor is that it associates, when it changes its shape, it associates with the G protein, and that is a trimeric protein, and that changes shape. And what happens is you get a cascade of events. Once the G protein is activated, you get a series of other proteins that become activated in what's known as the signal transduction pathway. This occurs inside the cytoplasm. So it involves secondary messengers, it involves calcium, it involves cyclic AMP, it involves molecules that, while in the cytoplasm, can amplify the message, and then the cell can respond most efficiently. And so that's what this video is going to attempt to discuss. That is, it's going to try to emphasize signal transduction pathways. And so, as I was saying, it's a cascade of molecular interactions in the inside of the cell, but it starts from a signal from the outside, and that's picked up by a receptor. And then the signal transduction pathway, which involves a few steps, will amplify and regulate, as we shall see, the cellular response. And so let me give you just a simplistic, believe it or not, a simplistic example of this. And so when the receptor picks up a message, maybe the particular message is a growth factor. Okay? So this is picked up, and now one thing I want to mention about receptors is they're very unique. And so they're kind of like an enzyme, if you can relate to that. So the particular active site or receptor site is unique to the particular signaling molecule. Once that reception takes place, it usually causes the protein to change conformation, and that will allow something to take place. And so this is what we mean by the 3 steps of signaling transduction pathway. It's reception, transduction, and then a cellular response. And so what usually happens is that a molecule changes shape. It's often an enzyme, and that particular enzyme becomes active. And so if it's a kinase enzyme, and I'll go over this in a moment, if it's a kinase enzyme, it's going to transfer a phosphate onto another protein in a process called phosphorylation. And that usually changes the shape of that protein, which then changes the shape of the next one, and the next one, and the next one, and the next one. And these things increase in concentration, therefore amplifying the signal. So just a little bit of message goes a long way inside. And you're like, well, then what's going to happen? Oftentimes, what will occur is that a molecule will go inside the nucleus of the cell. And basically, this gets detailed, but it's pretty cool. It's not the real topic of this video. But these molecules can interact with transcription factors. In other words, by phosphorylating a transcription factor, that engages a particular promoter region of a gene, which is where RNA polymerase binds, and therefore a gene is then turned on, or maybe it removes a molecule that's inhibiting a regulatory protein. So it can either turn on or turn off a response. And so if the gene is turned on, let's just say, then RNA polymerase will transcribe that gene into messenger RNA. So a lot of RNA is then produced. It goes into the cytoplasm, and then that cell's ribosomes will make a whole slew of enzymes that weren't being made before. And you're like, well, what's that going to do? Well, that's going to respond. The cell's going to respond to that. And so there's some pathways that are going to be turned on as a result of that. Perhaps the cell initiates division as a consequence of receiving this message. And so the transduction pathway is what we want to focus on today. And it's usually multiple steps. And when it's multiple steps, that affords an amplification, and it also affords regulation and better coordination internally. And so let's take a look at that. So when you think of molecules relaying messages from a receptor, these are mostly proteins that we're talking about in the pathway. And so they're kind of like dominoes. When one thing is hit, the next one changes, the next one changes, the next one changes. And so when one protein is activated, then that can become a molecule that activates other molecules. And then when those molecules become active, those can activate other molecules. And so it not only is it sort of a cascade, but it also is an amplification. So I want to emphasize this point because not only is it important, I find it particularly cool and useful. And it explains a lot about what transduction really is trying to accomplish, which is amplification. So let's say, for example, we're talking about the fight or flight response. And so somebody's chasing an animal, and epinephrine is released from the adrenal glands, and it's targeting a muscle cell or a liver cell. And the goal is, the response is to get the polysaccharide glycogen to release sugar in the form of glucose to raise blood sugar levels so that the animal can respond and run away. And so how that is accomplished is that the receptor protein picks up the epinephrine, and what that will do is change the shape of the receptor protein and basically activate the inactive G protein, because this particular receptor is a G protein coupled, so coupled meaning associated with receptor proteins. And so when that becomes activated, many of these G proteins become activated, and as you can see right here, one molecule, this is what I want to emphasize, of epinephrine can produce 10 to the second power of activated G protein, so that's a big amplification. Then this activated G protein will activate and adenylate cyclase. And this is an enzyme that's also membrane bound. And don't get caught up in the detail, but once that becomes activated, this molecule will then be able to convert adenosine triphosphate, ATP, into a copious amount of cyclic AMP. So it's the removal of the pyrophosphate, yada yada. But the cyclic AMP acts as a secondary messenger, And so what is that doing? Well, cyclic AMP now left 10 to the 4 power, so the concentration of cyclic AMP is rather large in the cell compared to 1 epinephrine. So check that out. So then cyclic AMP is capable of activating these protein kinases, which are plentiful enough in the cell, but they're inactive. And then when they become active, the kinase is an enzyme that is capable of transferring a phosphate. So kinase A gets activated, and then that activates another enzyme called phosphorylase kinase. So phosphorylase kinase becomes activated, and look, the numbers are increasing even more, 10 to the 5th. Okay? So 10 to the 5th. And then what is this doing? The inactive, that phosphorylase kinase activates, this is the crucial enzyme in what is going to be involved in the response. The enzyme that we really want going on in the cell is glycogen, which is a polysaccharide phosphorylase. So what does that enzyme do? That's capable of hydrolyzing or cleaving glucoses off of glycogen. And so that enzyme becomes active, and that's the point. And so look at this. So many of those enzymes. And so therefore glycogen is just going to get mowed up. It's going to get chopped up. And then the consequence is look at all of that glucose. So the cell is just spewing with glucose. It's just pouring out 10 to the 8 molecules of glucose 1-phosphate. And so that ultimately will diffuse out of a cell and raise blood sugar levels. And so pretty cool. So you get a ton of glucose being released from just one little message. So signal transduction, response, but ultimately the signal was amplified. So each step of signal transduction is sort of the transduced part of it. Usually there's different exceptions to this. It usually involves some sort of shape change. And so just to emphasize this, if I have sort of a molecule like this, and it's a protein, and it's what we call inactive, usually what this involves is some kind of, when a phosphate is stuck to it, this is a negatively charged functional group, it usually results in activation. And so what's happening here is that the protein is literally, and I'll try to emphasize this here, it's literally becoming something useful. And so it's now capable of doing something. And what's really cool about this is sometimes we want enzymes to be active, sometimes we don't want it. So it goes both ways. You know, like, it's not always appropriate to activate. Sometimes it's appropriate to inactivate. And so along those lines, what we're talking about is 3 things. We're talking about reception. We're talking about how proteins become activated right here in the signal transduction, not only activated but amplified, as we saw in an example. And then there's some sort of response. And so the cell's doing something, and that's the whole point. So it received a message, and then it's going to do something. Or it's going to stop doing something. And that's important, because regulation doesn't mean increase. Regulation means both increase and decrease. So really dial in on that. And so there's really two pathways involved if you're going to get something going or stop something. It involves the sort of stick a phosphate on or remove a phosphate. And so what we call this is phosphorylation is the addition of a phosphate group. Phosphorylation is adding it. And usually the protein enzyme that transfers the phosphate is a protein kinase. If you're unfamiliar with this, become familiar with it. It's pretty crucial. Hyperbole alert. It's one of the most important enzymes in the cell. So it transfers phosphates often from ATP, which is a good reservoir of phosphate, onto a protein, and then a process called phosphorylation. Now, to remove it is dephosphorylation. Okay? So there's a different enzyme that does that. It's a phosphatase. So a protein phosphatase removes a phosphate from a protein, so it's dephosphorylation. It's a mouthful. And so it acts as sort of an on and off switch, depending on what you want. So it isn't always that when you add the phosphate, it's turned on. Sometimes when you add the phosphate, that means it becomes inactive. So I wouldn't rely on that. But nevertheless, it's when you add the phosphate, it's a kinase. When you remove it, it's a phosphatase. And that way, cells are capable of receiving messages from the outside. And I'm not just talking about human cells, eukaryotic cells, I'm talking about bacteria as well. And so this is remarkable. This signaling transduction pathway, if it's found and it's conserved within all of these phyla, and I might add even kingdoms and domains, it's strong, compelling evidence from a common ancestry that all life on the Earth has evolved. And so it's really, really cool. And so again, believe it or not, this diagram is a simplification of the signal transduction pathway. But it's basically like dominoes, something like that. So it's the molecule is altered, which then takes an inactive kinase and makes it active. And then the kinase, as you recall this from a moment ago, is an enzyme that is capable of transferring a phosphate. So it grabs a phosphate from adenosine triphosphate, sticks it on the protein kinase 2, which then makes that activated because it has a phosphate, so it changes its shape. And then that, in turn, activates another kinase. And in so doing, it's increasing the numbers of these secondary messages, and therefore ultimately resulting in a cellular response. Okay, so let's get into these secondary molecules a little bit, because I mentioned these kinases, but there's some other small molecules in ions that can act as secondary messengers as well. So this is pretty cool. So the extracellular, that means on the outside, is some kind of ligand. Now, it could be a growth factor, it could even be a neurotransmitter, it's something, it could be a hormone, and that binds to the receptor on the outside of the cell, and that's usually called the first messenger. Because, again, I don't want to get to be like, well, here's a discussion of secondary messengers, and it's like, well, what was the first message? The first message was the signal molecule. And then when you're in the cell,